Today I'm very excited to be talking about this project that is called Micro to Macro. And that project was a partnership between San Diego State, Sunken Seaweed primarily, which is a company from dear friends of mine up in Northern California now. And we are going to be focusing on talking about the seaweed microbiome part of that project. So it all started with a beer in my backyard here in Ocean Beach, San Diego. Me, Tori, and Leslie, they're the seaweed farmers from Sunken Seaweed. We were, at the time, looking for grant opportunities, looking for funding as scientists do. And Leslie brought up the idea of applying for this very much out there NSF grant, very different kind of NSF grant called the Convergence Accelerator. And I think that's how a lot of science starts and a lot of great projects starts. It's just, having a beer with your friends and chatting about data and crazy ideas. And at the time, Leslie and Tori were working in partnership with Dr. Edwards from the beer pigs, the benthic ecology experimental research, phycology in general. If you know the group, you know that they're all over California. This is the San Diego chapter at SDSU. And so it started with the three of us chatting about this new grant. How could we converge our expertises? At the time, I was about to finish my PhD on coral microbiomes. And I was getting more and more interested in seaweed. I had already a little bit of experience in diving in kelp forests and collecting microbial data from kelp forests. So we put the idea down, talked to Matt, and Matt was very excited to get into this endeavor. He joined us, and we, of course, we couldn't have done this project without his expertise, and he was the PI of the project. And this is the NSF Convergence Accelerator. At that time, that was for the 2021 cohort. We applied for the track that was called the Blue Economy track. So they were looking for projects that were very multidisciplinary, big integration among different disciplines and teams, partners from the business sector, from the academic sector. And they wanted us to come up with something that was user-inspired. So different way of doing science, of thinking already how people are going to benefit from the deliverables of the project. And our project was one of the 16 awardees in our track, in the Blue Economy track. And the way that this grant works, it has two phases. Phase one, which is when we have a bigger cohort, and we are part of that cohort at the time. And we have 1 year to develop the project, to make more partnerships. And we also have to go through training on designing prototypes, pitching business ideas. And towards the end of that year, we submit another proposal for phase two and give a pitch delivery to NSF to see if we're going to get more grant money. So spoiler alert, we only got all the way to phase one, which to me was already huge, and I had a great time with this project. But if you're interested, check out this grant. It's a very interesting kind of grant, very different from the general NSF's grants. And the projects that ended up being awarded, that were our colleagues, our peers from phase one, it was just 3 out of, I think 3 or 4 out of the 16. So it was pretty competitive. But I highly recommend everybody checking out the projects that were awarded to phase two. But before anything else, let's talk about what we did in phase one. So our project was called Micro to Macro, and the idea was to develop a seaweed farming platform to support the grower from seed to sale. And we were focusing on the West Coast of the US because seaweed farming in the US. In general, it's still at its infancy. It's booming on the East Coast and in Alaska as well, but generally speaking on the West Coast, it's still beginning to pick up momentum. So our idea was to boost the industry, especially here on the West Coast. So that was the core team at the time. Dr. Edwards and Tori Polizzi as PIs, and myself as seaweed researcher, microbial researcher, and Leslie as seaweed farmer and researcher at the time. So our idea was thinking of ways of scaling up seaweed farming on the West Coast. And we had a two-pronged approach. The first one is to make farming accessible. So we're aiming to promote seaweed farming in policy ways, permitting ways. We're looking into streamlining barriers to entry. We were thinking of developing a training for new farmers. But that's not the part that I'm going to be talking about today, just so you know that that was a bigger project, and I am going to be focusing on the seaweed microbiome part of the project. And so the seaweed microbiome fits within the other goal, that is to look for ways, innovative ways to optimize crop yields. I apologize for the airplanes. If you've been to Ocean Beach in San Diego, we're right under the flight path here, very close to the airport, and it's a very busy, tidy airport that we have. So those were the objectives for phase one. We had farmer networking part of the project that was mostly led by Tori Polizzi, and the seaweed science, that's where I had more agency under. So thinking of seaweed beneficial microbiomes, our idea comes in a time where this is being widely talked about. It's very new, but at the same time, the potential benefit that microbes can grant to seaweeds is being very explored at the moment. It's very exciting to see papers. Every year we have so many different papers about the subject coming up. So we were inserted for sure in a cohort that has momentum. And the idea here, for example, is to harness the potential that native seaweed microbiomes have, that they already are already benefits that are providing to the seaweeds naturally, and to be able to manipulate that, especially for increasing nutrient acquisition and for allowing the seaweed to grow better and faster. That was the overall idea. And if in the pipeline of developing these potential probiotics or beneficial inoculums to seaweed that could boost seaweed farming, we have to start all the way from microbial sampling, so sampling as many seaweed microbiomes as we can, and then analyzing the data to filter through what kind of potential taxa and functions we're going to be targeting. And further steps are going to be related to isolating these taxa and specific functions, and running a bunch of different experiments on how the inoculums would be acquired by the seaweed, under what conditions, for how long, and if you would have to re-inoculate them. And that is a much more complex part of the process. So that's the general idea and the general pipeline to get to the seaweed probiotics. But in phase one, what we did in that year, we sampled more than 150 microbiomes all over the West Coast, California, Oregon, and Alaska. And we sampled from 7 different seaweed species, 3 seaweeds, and I think 4 kelp species. And we did that by using this device that Dr. Forrest Brower's lab developed a while back. That's called the super sucker, the infamous super sucker. It's this blunt syringe that you're seeing on the right side that it's manufactured specifically for that. You can't buy this gear just yet. But it's essentially a recirculating syringe that you load it up with filtered seawater. And as you flush it against the surface of the host organism that you want to sample the microbe from, so this is the surface microbiome we're sampling. The water gets flushed on the surface of the organism, and these two tubings that you're seeing on the sides of the syringe are going to capture the water and flush it back to the back of the syringe. So as you're flushing the seawater against the surface of the seaweed, the water is being recaptured and recirculated back to the back of the syringe. So you have a diluted sample of the microbes on the surface plus the seawater, the filtered seawater that you had in the syringe already. So that's what we used. And we sampled a few farms and also natural beds. And the way we would do it, especially in farms, we would clip the tissue from the lines and either sample them right away on the boat when that was possible, or bring them to as fast as we could to the lab to have the tissue sampled. And that liquid sample, that diluted sample, that mix of seawater and surface microbiomes, was flushed against a Sterivex filter. That's what you see on the bottom right part of the figure there. So you trap the microbial cells in that filter. And from that little filter, you extract the DNA and go through the whole process of the metagenomic analysis. Metagenomes means we're getting the genome from the whole community. So we're interested in knowing what are the microbiome as a community is doing, not just one specific taxon. And from the extracted DNA, we did whole genome sequencing using shotgun sequencing. We used the Illumina NovaSeq platform. And after we got the sequences from the sequencer, we would go through a bioinformatics pipeline. We annotated our metagenomes through MGREST using RefSeq and SEED databases for taxon function. And then from MGREST, you can get something that any ecologist is very used to, which is basically a community relative abundance data sheet that you can then run statistics on. So our data, part of our data is already public on MGREST, this platform. So if you get to MGREST and Google and search micro to macro, you will find our project there. We already have, I think, about 30 metagenomes in there and more to come. The thing is, MGREST sometimes it's not available at the moment. I think they're changing their server or something. So we have some samples that could not be processed just yet, but because the server has been not very reliable at the moment. But it is a great resource and a great way to annotate and analyze samples. So from this project, we envision having 4 publications resulting from it. And today I'm going to be focusing on 2 parts of this project, which is, the first one is the seaweed microbiome and nutrient uptake. And the second part is the microbiome of kelp farms in California. So beginning with the seaweed microbiome and nutrient uptake, we chose Ulva lactoca as our model species because they are a promising bioremediator. The picture here is Leslie from Sunken Seaweed when they had ulva growing in their lines. And also the picture on the left that you see is their tumble culture at the Marine Lab at San Diego State. So ulva grows really fast, really well. It's very easy to deal with. And also they're very tolerant to many different kinds of environmental fluctuations, including low salinity. So if there is a need for bioremediating freshwater runoff going to a bay like San Diego Bay, Ulva lactuca can be a great candidate. So that's why we chose that species for testing how they respond to different nutrient conditions, how the microbiome and the seaweed respond to different nutrient conditions. So to answer that question, we ran a quick experiment. We put about 20 grams of ulva in replicated cone-shaped culture tanks. We replicated each treatment with 2 tanks, and we had the high, medium, low, and the control treatments. We started the experiment and collected the surface microbiome on day 0, right before we exposed the seaweed to the treatments. And by the end of the 7-day experiment, we resampled the same seaweed, but now they were already distributed across different treatments. So we have one initial control treatment, and then we have the same control treatment sampled after 7 days, plus the nutrient treatment the different nutrient treatment concentrations that we also sampled by the end of the 7-day experiment. We used a fertilizer solution using Peter's Professional. It's a very common fertilizer, garden fertilizer, that has equal parts of added nitrogen, phosphorus, and potassium, if I'm not mistaken. Temperature was controlled at 15.5 Celsius. It was a tumble culture system, and we had 12 to 12 hours of light and dark cycles for the light period. So what does the microbiome look like on Ulva in general from this experiment before getting into the treatments just yet? The first thing that we see here is the most dominant taxa of bacteria on Ulva, and they are Pseudomonas, Rosiobacter, Pseudomonas, Rugeria, Gromella, Flavobacterium, Erythrobacter. One type of flavobacteria that we couldn't get to the genus level, Polaribacter and Meribacter. Those are the most dominant one. They make up to about 40% of the relative abundances of the seaweed, the Ulva microbiome. And here I want to highlight the Meribacter being one of those very abundant taxa, because Meribacter is known to promote cell differentiation on another species of ulva. So it has a very key role on ulva species. Some bacterial strains of that genus are essential for the correct development and morphogenesis of some ulva species. That hasn't been proven for Ulva lactuca specifically, but it's exciting to find that same genus in that sample, in those metagenomes, and gives us some clues about what could be happening to Ulva species in general. Now, focusing on the effects of our nutrient treatments, we ran a Permanova, which is multivariate analysis to show that there is a significant effect of the nutrient treatments on the microbiome structure associated with ulva. Each one of those dots represent a whole microbiome community of each, and each dot is it's a replicate. So for each treatment, we have 2 replicates, 2 metagenomes. So this is a multidimensional scale representation of the graph. So the closest you see these dots, the more similar the microbiomes are to one another. And you can see that the red dots are the high nutrient treatments microbiomes. They're more spread out away from the low and medium and then control treatments. And among those, the microbiomes that I showed you, most of the taxa didn't have a difference in their relative abundances when you compare them across nutrient treatments. Out of the 560 something total taxa, I think it was about 40 that had a significant difference in their specific relative abundance when you compare the treatments pair to pair. And out of the 20 genera that were lower under high nutrient treatments. So nutrient treatments never statistically increased any abundance of any specific taxon, but it decreased. It had a relationship to decreasing some of the taxon. And so out of those 20 that were significantly lower under high nutrients, 16 belonged to the order Rhizobialis. And here on these graphs, I just have a few because we had a plot that has 16 of those comparisons. Here I just have an example of Labransia, Cynorhizobium, and Mesorhizobium of some of the Rhizobialis that were among those 16. And the reason why that's exciting and that order is a super interesting one to see a relationship in this case is because you see on the schematics on the right side, those are the same order of microbes that are associated with legumes that can fix nitrogen for plants, for land plants. And it looks like they play a key role on seaweed microbiomes as well. So you have something analogous between land plants and sea plants in this case. And when we look into microbial functions, we also compared different microbial kinds of microbial genes that are associated with different metabolisms. And since we increased both the nitrogen and the phosphorus concentration when we added the fertilizer to the water, we looked into what the nutrient treatments did to those specific metabolisms among the microbiomes. And what I found really interesting and really cool to see is that on figure A, you can see that the relative abundance of nitrogen metabolism genes for the seaweed-associated microbiomes increased under high nutrient concentrations. And you see that the controls, which are the gray colors, and the low to medium nutrient treatments are all among the same range, but under high nutrient conditions, we have a peak of an increase in microbial nitrogen metabolism genes. But that didn't happen to phosphorus metabolism. There's no clear trend or relationship there. And it looks like that under high nutrient conditions, the microbiome of seaweed responds very promptly to nitrogen, but not to phosphorus. Doesn't seem to be shaping the microbial metabolism in this case. And so zooming in into the nitrogen metabolism, not only they had more nitrogen metabolism genes in general under high nutrient conditions, but the seaweed microbiome also had different types of metabolism of nitrogen under high nutrients. So under high nutrient concentrations, they had less ammonia assimilation genes in the microbiome. And on the other hand, they had more denitrification genes. So what does that mean? We believe that this is the schematics of the nitrogen cycle. And the top part is when the nitrogen is transformed under oxygenic conditions. And the bottom part is the reactions that happen under anoxic conditions. When you see the ammonia here, Ammonium is the preferred nitrogen form for the seaweed to uptake, for ulva specifically. Kelp, giant kelp prefers nitrates, but ulva seems to prefer ammonium. They also love nitrates, but have a preference for ammonium. So they're very good at taking up ammonium from the water column. They assimilate that very fast, especially, it seems like, under high nutrient conditions. And secondly, nitrates. And microbes in general also prefer to assimilate ammonium. But we saw that ammonia assimilation decreased under high nutrient concentrations. And on the other hand, we had an increase in denitrification genes in the microbiome under high nutrient concentrations. So it could be that the seaweed is uptaking ammonium very fast, and the microbiomes is having to change their strategy and how they're going to use nitrogen, because it's quickly being uptaken by the seaweed. So the takeaways. First one is that Meribacter is a key microbe to ulva mutabilis and is also a dominant taxon in our data set, which is for further consideration, further directions to be looked into. Nutrient concentrations could be modulating the effectiveness of inoculums. I mean, this is more of a thinking about, because different nutrient concentrations can change the microbiome and how they're even metabolizing things. This is something to keep in mind for the future inoculums for seaweeds, because that could affect how the seaweed is uptaking the microbes that we're exposing them to and how the beneficial microbes are going to interact in that scenario. So keeping in mind the nutrient concentrations are going to be very key in future developments of seaweed probiotics. Also, one hypothesis is that ulva lactuca could be outcompeting some bacteria via the fast uptake of ammonium and nitrate. Again, this needs to be further explored. This is just a hypothesis coming from our data. Very exciting to think about it, to see that that could be happening. And rhizobiales stand out as key to both land and ocean agriculture. And among the rhizobiales, specifically Librantia, it had a negative relationship to growth of Macrocystis perifera in a different study. And here we also show that kind of relationship too with a different seaweed species, which is very exciting and important to be looking into these greater mechanisms between different species of seaweed and their microbiome. So let's take a breather, because I know talking about microbes can be a lot. And let's just admire some of the pictures that we took during my PhD when I was doing research on coral reefs. And the reason why I'm talking about coral reefs right now is because I love ecosystem engineers, and I have a huge passion for corals. And that passion was transferred to canopy forming kelp forests that we have here in California. So that was my smooth transition from coral reefs to kelp forests, and still looking into their microbiomes, because a lot of times you also forget that microbes are huge ecosystem engineers as well. They also form the foundation of our beloved coral reefs and kelp forests. And I'm from Brazil, so I thought when I moved here in 2016, I had never seen a kelp or a kelp forest before. We don't have those down there. And I thought it was going to feel very out of place coming from a very tropical environment to a temperate environment. But as soon as I dove in the kelp forest for the first time, I just felt super at home. It took me a while to get used to the wetsuit, the thickness of the wetsuit, but the kelp itself was fine. I loved diving in them, and they're still one of my favorite ecosystems. So showing our appreciation for kelp forests, now we're going to talk about the microbiome of kelp farms in California. And the farms that we have here in California are just starting. So we have a few farms of giant kelp and bull kelp. And it's a very interesting time to be sampling their microbiome and learning how the microbes are responding to these different systems, because it's an exciting, very brand new beginning of an industry here. And the picture on the left shows one of the closest ways we can get to actually see microbes. So all the colored dots, are the different colors, shows different taxa of microbes. And that is a photo of a kelp blade, a bull kelp blade cut cross-sectionally, just so we can have a little bit of a visual of the kelp microbiome before we get into data. So the question that we wanted to answer here is, how does the kelp microbiome vary spatially and temporally? And we had just 1 year to sample, so it's not like we're going to have many replicates of different seasons or even different kelp farms that we could go to several times in a row. So this was a very opportunistic sampling effort. And of course, there's a very specific dynamic of when the kelp is seeded and the lines are putting out in the water to be grown in the farms, and when they're harvested. So we also had to work with that seasonality as well, of things being harvested at a certain time of the year. And when they put the lines back in the farms, a lot of the times there's still tiny sporophytes or tiny gametophytes. So just keep in mind that this is going to be a very broad view of playing naturalist with microbes in these kelp farms and some natural beds as well. So the opportunities that we had to sample was, specifically in California, was sampling for Macrocystis giant kelp in Santa Barbara in fall 2021. We sampled the Mohawk Reef, is a natural bed, and the NO4, that's the 4 metagenomes that worked. We sampled more samples, but with molecular biology, if you know, you know. A lot of times we do exactly what the protocol asks us to do. Things still don't work. Sometimes the sequences are just not very good when they come out of the sequencer. So we always end up losing samples in the process. So we got 4 good metagenomes from Mohawk Reef, 5 from Ocean Rainforest, our dear collaborators from Santa Barbara. And we also sampled bull kelp in Northern California. And we did that in the winter in the natural beds of Trinidad Head and Humboldt and Van Dam State Park in Mendocino. And that's where we clumped all of that as natural bed. I know it's not technically the same site, but the microbiomes were pretty similar, and we felt confident to put them in the same category of natural beds for comparison. And the farmed bull kelp microbiomes were collected from Green Wave in Humboldt Bay. And that one was cool because we could go actually in 2 different time periods, in the spring 2022 and then back to the very same farm in summer 2022. And by the way that I showed in the beginning how we sampled the microbiome, that's how we did for the Ulva project and also the kelp. It's the same methodology. So again, the total richness of the bacterial communities associated with these 2 kelp species was more than 500 genera. Here, I only have the dominant ones, and we're going to talk about a little bit about their potential roles according to the literature. So Pseudomonas have been associated with nitrogen acquisition in some seaweed species. Rosiobacter has been associated to morphogenesis for certain seaweed species as well, specific strains. Schwannella is involved in carbon degradation and associated with biofouled seaweeds. Rhodopyralula seems to be very important in the DOC consumption in kelp forests, so carbon degradation also. Flavobacteria and Gramella, Flavobacterium, and Ducdonia are all flavobacteria. All of those three can be involved in alginate degradation. Akkermansia can produce antimicrobes, and fun fact, they're also a human gut probiotic. So they're very good at digesting mucin in our gut. And for seaweeds, they're good at degrading alginate, and they can produce antimicrobials as well. Vibrio, a lot of the times, Vibrio can be associated to pathogenic roles, but they're also even used in bioengineering for degrading alginates. They can be bioengineered for the specific genes, and also associated with biofouled seaweeds, which a lot of the times we never know if is it the biofouling that's causing more carbon degradation, so we're seeing more of those microbes associated with them, or do the microbes have any role in facilitating the blades to be more fouled or not? This is still something that needs to be further investigated. Rugeria has been associated to seaweed growth and defense. Rickettsia associated with biofouling, and Pseudotermonas associated with agar degradation and antimicrobial production. So generally speaking, many microbes that are related to carbon metabolisms. Some help the seaweed to acquire nutrients or recycle some nutrients. Some have been associated with not very desirable, not so desirable features such as fouled blades. But that's the general picture here. Now we know a little bit about each taxon and what they could be doing on the seaweed microbiome of these two kelp species. And looking more now into the similarities or differences across these different groups, this again is a principal coordinated analysis. So each dot here represents one replicated microbiome, so a whole community in each dot. And the closest you see those geometric figures, the more similar they are. The orange ones are representing the giant kelp microbiomes, the brown ones, the bull kelp. And I also added just 4 different water microbiomes as well, microbial communities. We have many water samples. It's just very costly in terms of bioinformatics to add a lot of extra samples. So just for reference, I added some water microbial communities, so you know that they are actually very different from the kelp microbiomes. The big pattern that we see here is that kelp farms have an effect to homogenize the microbiome that's in them. For example, the greatest similarity that we had within groups, within the kelp farm group in the summer, we had a 91% similarity, meaning from all the metagenomes that we had for that group, they shared 91% of their microbial structure. It's pretty high. And second to that was the giant kelp farmed microbiome. And in third place, also a kelp farmed microbiome, but in the spring, followed by natural beds of giant kelp and then natural beds of bull kelp. So it's not surprising that if we expose the kelp to a very homogenized condition, that they're going to have a more homogenized microbiome. It's similar to land plants as well. But zooming in, because we had such a scattered way of sampling, like I said, was very opportunistic. We only had 1 year to actually just less than a year to accomplish the sampling. We couldn't make very good comparisons between groups because we didn't have, for example, two seasons replicated for each species. But we had farm versus natural within giant kelp, and that was done in the same season, so we're ruling out the seasonal effect. In that case, giant kelp from farmed settings versus natural settings, their microbiome was dissimilar about 15%. So they were similar around 85%. And statistically, they showed different microbiomes. It all depends at the scale that you're comparing things, if they're going to be statistically significant or not. If we're zooming in and comparing farmed versus natural within giant kelp, we see a statistic difference there. In farm settings, we had more of the Akkermansia genera and Rhodopteralula, and in natural settings, we had more of the Schwannella and the Vibrio. And it could be that the natural seaweed microbiomes and the seaweed microbiomes in the natural kelp beds, because the kelp beds are more foul, they're more variable, they're more exposed to the elements, it could be that we have more Schwannella and Vibrio for that reason. And now zooming in and looking into bull kelp farms, we could compare spring versus summer because we sampled the same farm in these two different seasons. And the similarity was actually greater. So the same farm sampled between two time periods had a greater dissimilarity than here when we're comparing a farm setting versus a natural setting. So different pools of species and individuals versus here, the same group of individuals sampled temporally had a greater dissimilarity. It could be that because this was sampled from Humboldt Bay, there's just more dynamic of nutrients and freshwater runoffs than it would have in a coastal setting, just like the giant kelp farm in natural beds were sampled in. In the spring, we saw more rickettsia on the bull kelp blades, and in the summer, we saw more of the flavobacteria. Rickettsia has been associated with biofouling in other seaweeds. It could be that in the spring, with the nutrient runoff, they got more of the invertebrates growing on them. But that's still, we need to further investigate these things. All right, so the major takeaways about the kelp farms in California is that the farms, generally speaking, homogenize kelp microbiomes. And the microbiomes, even with the differences that we're seeing statistically in the percentages and the similarity, the microbiomes are still very comparable to the natural bed. So it's not that the kelp farm creates a dysbiotic state where the microbiome is completely not functional or leading towards an unhealthy state. It doesn't look like it. It looks like the kelp farms can produce very healthy microbiomes as well. They're comparable to natural beds. Natural beds of different kelp species host similar microbiomes. So when we compare all of the groups that we had from giant kelp, bull kelp, farmed, not farmed, different seasons, the natural beds, the kelp microbiomes of natural beds from the different species were relatively similar when we take the whole context in consideration, which is also very exciting to see that the natural conditions are creating similarities in the microbiomes that go across beyond specific species, and that the farms can create more specific microbiomes related to them. Farmed bull kelp microbiomes showed pronounced temporal changes. So it would be really cool to go back to the Green Wave Farm and sample that temporally, or even the Ocean Rainforest Farm, and then follow a temporal data set and see how the microbiome can change seasonally. So the Micro to Macro project is a wrap. It's done. I literally just finished my postdoc with Dr. Edwards. And what we did was to create a baseline data for microbiomes from farmed seaweed. And again, we still have a fair amount of samples to make available to the public as soon as the server gets back to running properly. And we identified some potential seaweed beneficial taxa that can inform further efforts. So we fit right here on the pipeline to the right, where the part of this pipeline of developing seaweed probiotics, we helped to inform where the next direction should go, and provide a big database that anybody can access, and then be more specific about the microbial taxa or functions they're looking into. I hope to see you at the California Seaweed Festival. That's coming up. It's in San Diego. I'll be giving a different version of this talk. It's going to be very similar, but I'll be moderating that session. So I hope to see you there, and let's chat in person. I always like to connect with new people. And if you want to connect with me right now, I didn't add my email address because I'm changing institutions. But if you go to my website, I will have my most up-to-date email address there soon. And thank you so much. I hope you enjoyed the talk, and please let me know if you have any questions or any insights.